for this second plenary session conference. On behalf of the organizing committee, thank you very, very much, Professor, for accepting our invitation. Before starting this conference, let me just briefly introduce Professor Juleen Zierath and summarize her outstanding career. After a PhD at the Karolinska Institute in Harriet Wahlberg and Eriksson's lab, and a postdoc in Harvard Medical School in Barbara Kahn's laboratory, Juleen moved back to Sweden. She is now Professor of Clinical Integrative Physiology at the Karolinska Institute in Stockholm and head of the section of Physiology at the Department of Molecular Medicine and Surgery and the Department of Physiology and Pharmacology. She is also Executive Director of the Novo Nordisk Foundation Center for Basic Metabolic Research in Copenhagen, in Denmark. And Juleen Zierath leads research groups in both Stockholm and Copenhagen. Her research has provided key results in the cellular and molecular mechanisms underlying glucose homeostasis in skeletal muscle and in their alteration in physiopathological disorder like type 2 and diabetes. Another important issue of your work, I believe, is to translate basic research results into beneficial medical practices for patients, for instance by promoting physical exercise for prevention and treatment of metabolic diseases. You published more than 300 articles, it's very impressive, and many of them in high-impact journals such as Diabetes, Diabetologia, Cell Metabolism, you name it. Professor Zierath sits also on the Nobel Committee for Physiology and Medicine, and actually you have been chairwoman of this Nobel Committee between 2013 and 2015. Additionally, the international scientific community has recognized your exceptional achievement to research in the diabetes field by granting you numerous awards. And let me just mention a very special awards you received a few weeks ago, very recently, the prestigious Claude Bernard Medal and Lectureship Prize from the European Association for Study of Diabetes. So congratulations. Last thing I want to highlight, Juleen Zierath plays also important roles in influencing and guiding choices and strategies for current and future research programs in Sweden and also in Europe. So again, thank you very much, Juleen, for being with us today. And we are delighted to listen to your conference entitled Exercise, Inactivity and Circadian Rhythm, Resetting the Clock in Metabolic Disease.
Thanks so much for the opportunity to be here. I've really never been in this city before, and it's so lovely. And it's great to be at a face-to-face conference. I think we've all longed for that. All right, so my team is really interested in how our environment affects our metabolism. And you mentioned that I'm thinking a lot about the prevention of insulin resistance in diabetes. And we're interested in how environmental factors, what we expose ourselves to, can affect our metabolism. And for the purpose of this talk, I'll spend most of my time talking about how exercise, an environmental factor, something we can control, can steer our metabolism. And so I'm going to give you an overview of my lecture. So I'll talk about the effects of exercise and physical activity on metabolism. And I hope that by the time you leave, you can think that exercise may be a medicine that we can use for people with diabetes. And then I'll share some data that we have where we can remodel muscle to make the muscle cells more insulin sensitive. And I'll give you some examples of how exercise can really help the muscle and how inactivity is deleterious for the muscle. And then finally, I will share some new work from our lab where we synchronize the time of day with what we do. So can we synchronize our exercise intervention with the molecular circadian clocks? And I'll talk about some of that new work. So I think of exercise as really the ultimate integrative physiology. And this slide really recapitulates all of the things that exercise can do on multiple organ systems. It can be important for our nervous system, our metabolism, oxygen transport. But for the purpose of this talk, I'll spend most of my time talking about how exercise can be beneficial for muscle and metabolism. We do think about exercise as having effects on many, many organs within our body. We know that exercise can have an important effect on our lung capacity, our cardiac system, our metabolism. And again, I'm going to spend most of my time on muscle, but the whole body experiences exercise. So it's not just one organ system, it's multiple. So if you kind of look into a muscle cell, a working muscle cell, we can kind of get to the molecular mechanisms by which exercise has an effect. And if you look down here, we can look at either acute exercise or exercise training. And we can put this in a scale of hours, days, weeks, and months. And really within the first hours after exercise, the exercise leads to changes in the messenger RNA. And this happens almost acutely, but also within hours after the bout of exercise. And this mRNA bursts leads to changes in the abundance of proteins. Many of these proteins are important for controlling metabolism in the cell. And clearly with weeks or months of repeated exercise training, you have these functional adaptations that occur. There's improvements in work performance, there's improvements in metabolism, and much of this is a consequence of these repeated bouts of exercise daily or every second day that can ultimately change our physiology. So when I got into the field, I was really inspired by the work of my mentor, John Halazzi, and my PhD advisor, Harriet Wahlberg Henriksen. And what they did was they studied diabetic rodents, and they took the muscle from these rodents and they stimulated the muscles to contract, to exercise. And these rodents were insulin resistant and insulin deficient. So they were severely diabetic. And what they showed was that when they took the muscle from the diabetic animal and forced it to contract, they found a very robust increase in glucose uptake in these insulin resistant muscles. And they put the notion forward that exercise is a medicine. This contraction, the exercise, could increase glucose uptake by an insulin-independent mechanism. And so because of these findings, we've been really interested in understanding how exercise signals in a muscle cell to steer metabolism. And we do now know that different kinds of exercise, whether it's strength training or endurance training, can lead to specific changes in the muscle cell. We know that the resistance training can lead to muscle hypertrophy, and this occurs through a signaling cascade that's fairly well defined that involves PI3 kinase, AKT, mTOR, and such. Whereas endurance training leads to primarily changes in metabolism, and a lot of that's steered through mitochondrial biogenesis. So in broad strokes, we have an understanding that these two different modalities can modify the phenotype of the muscle. So now I'm going to get into some of the questions that we're raising in the lab. How does different exercise modalities reprogram a muscle cell? And so to do this, Nico Pilan in the lab created a meta-analysis of the muscle's response to exercise and inactivity. And what Nico did was he went into the literature and he found all of the muscle transcriptomic data sets that had been deposited. And he had approximately hundreds of studies and 700 volunteers where there were muscle biopsies that were taken across either aerobic exercise paradigms, resistance exercise paradigms, or inactivity. And in fact, if you write down this website, you can utilize the tool he built. And so he curated these data sets, and he wanted to ask, how does the muscle respond to either acute exercise, training, and do different modalities of exercise or inactivity change the muscle's fingerprint? And so he curated all of these 700 volunteers into their different training modalities.
And he could also segregate the populations based on age and body mass index.
And so he could really refine the subject pools. And so this is an example of a gene called PGC1 alpha, and that's a gene that's important for mitochondrial biogenesis. And what Nico was able to do was to take all of the different studies, and he could survey them in a meta-analysis for how does different training modalities affect the expression of PGC1 alpha. And what he could find is that with these forest plots, it was really the acute exercise that led to changes in PGC1 alpha, whereas the actual training exercise had a normalization of PGC1 alpha. And what's interesting is that here, where exercise boosts up PGC1 alpha expression, inactivity leads to a downregulation of that gene. So you can actually use this database to query your favorite gene for how it behaves in response to exercise.
Now this is a principal component analysis of all these data sets that Nico correlated, and you can see that the transcriptomic response of the muscle to acute exercise, whether it's resistance training or aerobic exercise, all those genes cluster together. And here are the actual training studies. So there's a separation of the effects of acute exercise from resistance training. And so then Nico could take all of these paradigms and he could look at the effects of these paradigms on the muscle transcriptomic. And what this slide shows is that there are very specific gene signatures that you can pull out in response to either acute training, resistance, acute aerobic training, inactivity, aerobic training for many, many weeks, or resistance training for many, many weeks. And so what this says is that different exercise perturbations lead to specific signatures of gene expression. Now the other thing you can do with the data set is you can ask whether or not specific metabolic pathways are altered in response to these exercise perturbations. So one can see here that with lipid metabolism, most of the exercise paradigms lead to a boost in gene expression, but inactivity leads to a downregulation of the genes. You can pull out signatures for cytokine response. That happens actually very early in response to acute exercise, and that is like a stress response in the cells. You can see a signature of fiber type switching in the cells, and that's mainly in response to the training. And this one here, mitochondrial responses, I'll spend more time in my presentation discussing. So what we can see here is that it's really the aerobic training paradigms that increase the expressions of genes that are associated with the mitochondria, the powerhouse of the cell. And conversely, inactivity dampens the expression of genes that are important for the mitochondria. All right, so the last thing I want to show you with this example from Nico's work is that one can even query the database for healthy versus metabolically impaired individuals. And you can make a comparison of genes, in this case to the mitochondrial respiratory change. You can see that all exercise, mainly aerobic, boosts up the mitochondrial respiratory chain gene expression. And we also, this is an important public health message, we get a boost of the expression of genes for mitochondrial respiration in people who have metabolic impairments. So the point of this slide is that exercise is good for people who are healthy, but it's also very good for people who have a metabolic disease. And with repeated aerobic training, you can actually boost up your mitochondrial capacity. All right, so there's specific adaptations to training. We can see that based on different phenotypes at the level of the cardiovascular function, at the level of the mitochondrial biogenesis pathways. And this is specific to the type of training, the aerobic training. And with resistance training, we have an increase in muscle hypertrophy, we have an increase in strength, and other enzymes that are important for lactate metabolism. And one thing we can say is that all training, whether it's aerobic or resistance, all training types promote increase in glucose uptake, glucose metabolism, and they improve glycemic control. So our work has given some resolution to the molecular machinery, but we also know that the physiology is markedly impaired because of the gene expression changes. All right, so this is a cartoon just to say there's still many mysteries. Nico's work has put some of the molecular machinery into perspective, but he's also been able to, through his work, identify new biology. And here is an example of genes that were changed in response to either aerobic exercise or resistance exercise. And what he looked for were genes that went in one direction with the training and the opposite direction with inactivity. And he found 4 genes that were activity-responsive genes. And in fact, one of these genes, a nuclear receptor 4A3, was a gene that we could confirm in a second cohort. And we also could show that in a plate of myocytes in muscle cells, if we stimulated the muscle cells with electrical stimulation, it mimicked the effects of exercise. So what is this gene? Nico identified it in his gene array studies, but there was very little known about its effect in metabolism. So Brendan Gabriel took this gene, this nuclear receptor gene, and he stimulated muscle cells in a dish in a dose-dependent manner, and he could show that the contraction of the muscle increased the expression of this gene. Again, a dose-dependent effect of contraction. He silenced the expression of the gene. He knocked it down. He made the muscle look like it was inactive by silencing the gene. And he could show that when he silenced this nuclear receptor, he actually could block the effects of contraction on glucose uptake. So it provided evidence that this nuclear receptor gene is important for steering glucose metabolism. And this is just to show you that silencing NR4A3 made the muscle look like an inactivity phenotype in terms of a whole set of genes that he identified. They have evidence that NR4A3 is an exercise-responsive gene, and it's also down-regulated with inactivity. So another feature of down-regulating the gene is that it blocks oxygen consumption rates in the cells, it blocks the glycolysis in the cells, it lowers the expression of genes that are associated with the mitochondria, and it affects the cells in that they become more quiescent. So what we've been able to do with this gene expression profiling is create a landscape of the muscle's gene profile in response to acute exercise, exercise training, or inactivity. And we've been able to come up with some new exercise-responsive genes that were not previously appreciated. All right, so for the last part of my presentation, I'm going to talk about this new area of science in our group and how can we synchronize what we do with our molecular circadian clock. And so what we now know is that nearly every cell in our body has a timekeeper, has a molecular circadian clock. And these clocks control the expression of thousands of genes. And in the brain, in the suprachiasmatic nuclei, lies the central clock. This is the master clock, and that coordinates the interaction with the environment to the other clocks in peripheral cells. So there is a clock in muscle cells, a clock in fat cells, liver cells, skin cells, all the cells in our body, nearly all cells have this molecular machinery. And this is a transcription-translation feedback loop. So what we know is that in response to light cues or other Zeitgebers, Zeitgeber is a timekeeper, nutrients, hypoxia, metabolites, secreted factors, hormones. In response to pulses of these Zeitgebers, there is an activation of this clock where we can then turn on the expression of many genes, and over time it turns off the expression of genes. So it's an autonomous clock that is affecting metabolism within cells. And what we're interested in is whether or not exercise may be another Zeitgeber, another timekeeper. And can exercise somehow fine-tune this clock machinery? And can it then affect the output of this clock? And this is mainly metabolic genes. All right, so this slide is a very complicated slide, but I use it as an example to show you how muscle cells have this molecular machinery. And so what Ali did was he took a plate of myotubes, human myotube cultured cells. So he took them out of the body, he cultured them in a dish, and he shocked them with serum. And that is a serum shock, and what that does is it resets all of the clocks. Because every cell has its own clock, the serum shock aligns those clocks so they're ticking, to say, in unison. And so what Ali did was once he serum shocked these cells, he was able to show these rhythms in gene expression and methylation. That's a molecular machinery that can control the expression of genes as well. And the most important thing in this slide is that many of the genes that are circadian in these muscle cells, they're associated with development of the muscle and also metabolism of the muscle. So it's an example of muscle cells have this circadian machinery. All right, so if we take a big picture approach here, together with the late Paolo Sassoni-Corset, John Hawley, Harriet Wahlberg Hendrickson, and Anna Croak, we've been asking whether or not diet and exercise can interact with this clock machinery. And there's a rhythm of this machinery throughout the day. So the question we had was, is there a more optimal time of day to exercise or to eat? And could you align that with some of this machinery? And I mentioned it's a transcription translation feedback loop, so could you hit this machinery when transcription factors are high? And if you could do that, would you be able to boost up the effects of exercise on mitochondria or metabolism? And so that's what we've been thinking. And we have actually some evidence from some animal studies where we take mice and we have them exercise either at Zeitgeber time 15 or Zeitgeber time 3. And this is either in the active phase for the mouse or the rest phase for the mouse. And we have the mice do 1 hour of exercise and then sacrifice the mice and study them over a circadian cycle. And what we found is that when the mice were exercising at the Zeitgeber time 15, at the active phase, it was only then that we saw effects of exercise on many metabolic pathways. So when the mice exercised in the rest phase, we really didn't see effects of the exercise on metabolism. So in this rodent study, published a couple years ago, we provide evidence that the time of day that the exercise is performed can have an effect on systemic metabolism. And we think that some of this is through the transcription factor HIF1 alpha. All right, so if we move into the clinic, we can ask the question, is there an optimal time of the day to exercise for people with diabetes to achieve a blood glucose control? And if you take a look over here, this is continuous blood glucose monitoring of individuals who are having normal glucose tolerance and individuals who have type 2 diabetes. And you can see already that there's a very, very huge effect on the glycemia. The people with diabetes clearly have hyperglycemia that's associated with each meal. And our question was whether we could exercise either in the morning or the afternoon to mitigate some of the peaks in the blood sugar throughout the day. And so we did a crossover study. We recruited 11 men with type 2 diabetes. We brought them into the lab, and they either performed exercise in the morning or in the afternoon. And they did a 2-week training study with a high-intensity interval training. And they performed the exercise Monday, Wednesday, Friday, and they did 6 times 1 minute bouts of high-intensity exercise. And we fitted these men with continuous blood glucose monitors, and we followed them. And you can see here we had a washout period, and then they came back and they performed the opposite arm of the training for 2 more weeks. All right, so the men were their own controls, and this is their blood glucose levels in the control state in gray, okay? And you can see that here between the different training regimes. So it's either week 1 training or week 2 training. And so if you look here at the blue, these are the men that perform the exercise in the afternoon. And you can see here that there's significant effects of the exercise afternoon training to lower the blood glucose levels. However, and surprisingly, when the men performed the exercise in the morning, there was actually an elevation in their blood sugar compared to their fasting, their running period. And in fact, the morning training led to an elevation in blood sugar. And we think that that might be related to catecholamine responses and a failure of the liver to turn off hepatic glucose production. So they really come in with high sugars and they're not able to lower it. So the message here is that perhaps the afternoon exercise may be more beneficial. The other thing we did in this study is we took muscle biopsies from these men in the morning, 36 to 48 hours after the last bout of training, and we took the muscle biopsies at the same time of the day. But the men had performed either afternoon or morning training. And we did a proteomic analysis of the muscle. And overall, what we found was that there were changes in muscle proteins, and many of those changes occurred in a time of day dependent manner. And what we found was that when the men performed the exercise in the afternoon, there were changes in genes that were, sorry, this is afternoon. When the men performed the training in the afternoon, there were changes in proteins that are associated with the mitochondria. But when the men trained in the morning, there were changes in the abundance of proteins that are associated with glucose metabolism. So the point of this story is that the afternoon training promoted mitochondrial biogenesis. The morning training promoted changes in glucose metabolism. Mladen also did a metabolomic analysis on these samples. And again, the samples were taken 36 to 48 hours after the last bout of either morning or afternoon training. And he looked at the plasma metabolites, the muscle metabolites, and the subcutaneous adipose tissue metabolites. And the idea here is that the exercise training did lead to changes in the metabolites in a tissue-specific manner. And what I can show you here is that there was correlations between the tissues, between the muscle, the plasma, and the subcutaneous adipose tissue for these different metabolite classes. And the bottom line of this graph is that the morning training was associated with more metabolites that are associated with glucose metabolism, and the afternoon training was associated with more metabolites that were changed related to lipid metabolism. And so there seems to be a theme that the different time of day of exercise somehow did rewire the muscle's metabolism and protein abundance. All right, so this is now unpublished work, but I want to share it with you. And it's part of a larger study together with Dominic Lutter and Ken Dyer. And it's going back to the work that was published in 2019 with Paolo Sassoni-Corse. And it's this model where the mice are trained either at Zeitgeber time 15, all in the green here, or Zeitgeber time 3, all in the blue. And what we did was we had the mice perform 1 hour of exercise, either at the early active phase here, or the early rest phase here. And we took all of these organs from the mice right after exercise, and for 4 hours, every 4 hours for 24 hours. And we did a metabolomic analysis. And clearly you can see that there's time of day effects of the exercise on the way in which these organs metabolize. If you just look here at the white adipose tissue, very few metabolites were changed when the mice were training during the early rest phase, but many more metabolites changed during the early active phase. So time of day seems to matter on how the machinery, how the muscles and how the organs perform metabolism. This diagram is to illustrate how a sedentary versus an exercise-trained animal behaves in response to exercise. And here we have all of the different organs that we studied from these mice acutely, either in the sedentary state or after exercise. And here we see correlations between the metabolite profiles of, let's say, a serum and heart, or brown adipose tissue and liver, for example. But what's striking is that when an animal exercises, there's many different organs that are engaged in performing that work. It's not only the heart, it's not only the muscle, but many organs contribute to the systemic metabolism. And you can see that there's correlations between metabolite changes in serum and liver, in muscle and liver, and heart. And some of the biggest connections are between the muscle and liver. And so what this shows is the relationship between the metabolite changes either during the early rest phase or the early active phase. And again, the mice all get 1 hour of exercise, but the time of day in which they exercise is different. And clearly you can see that when the mouse is exercising during the early active phase, there's much greater disruption of metabolism. There's many more metabolite changes that you see. between the muscle and the liver. And so part of this may be related to the substrates on board. So whether the animal is either fed or fasted. And in the case of a fasted animal, there's a greater metabolic disruption of homeostasis under those conditions. And there's a greater stress on the metabolic machinery. So time of day that the exercise is performed and whether or not the organism is fasted or fed can influence these results. All right, so I want to move now into the last vignette that I'm going to share with you today. And this is some work that has just recently been published. And again, we're still thinking about time of day and what we do and how that interacts with this molecular machinery to control metabolism. And primarily, does it affect mitochondria? And so what Brendan Gabriel was interested in was whether or not there was a connection between this molecular clock and our mitochondrial biology. And he wanted to ask whether or not the muscle mitochondrial function is under the control of this clock. And so what Brendan did was he took skeletal muscle from healthy individuals or people with type 2 diabetes. He then isolated satellite cells and he grew them in a plate. And he differentiated those satellite cells into myocells, myoblasts, and then myotubes. And then he serum shocked those muscle cells to reset the clock. And he extracted RNA and performed RNA sequencing. And he also performed a seahorse analysis where he looked at the rhythmicity of the mitochondrial oxygen consumption over the course of 48 hours. And you can see that the oxygen consumption rates are rhythmic. And that's very similar to what Patrick Schrawn has seen in muscle biopsies taken from humans throughout a day. You can see there's a rhythmicity in the oxygen consumption of the mitochondria. So Ali, who really likes big data, took all of the gene expression profiles and he did RNA sequencing, he did a RAIN analysis, and he could establish whether or not the changes in gene expression throughout this 54-hour cycle were rhythmic or not. And he compared these profiles between the type 2 diabetic, always shown in red, or the healthy individuals shown in blue. And this really summarizes what Ali found. He found that the myotube cultures from the healthy ones in the blue, many of the genes had a diurnal rhythm. They had a circadian rhythm. But when he looked at the cells from the people with diabetes, he found that that rhythm was dampened. So in many cases, the clock machinery, but clock output genes had a dampened rhythm. And in fact, that's what we do see in biopsies from individuals who have obesity. We have found that in muscle biopsies from people who have obesity, they have a disruption in some of the clock genes expressed in their muscle. We can show that the systemic lipid levels correlate with the abundance of many of these clock genes. And when we take myotube cultures from healthy individuals and culture them in the presence of palmitate, a lipid, we can disrupt the clock genes and the clock output genes. So if we use that and we take this model, we've got our myotube cultures, and many years ago, Karim Bouzakri and I showed that if you culture the myotubes from healthy individuals, you can get an effect of insulin to improve glucose uptake. But if you culture the cells from the people with diabetes, they actually have a memory of their insulin resistance. So these cultured cells maintain their insulin-resistant phenotype. So what Brendan did was he went back into the lab. He had his rhythm from his normal glucose tolerant cells. He had cells from healthy people, and they have a rhythm in their oxygen consumption rates. But when he studies the myotube cultures from the people with type 2 diabetes, he can see that the rhythm for oxygen consumption is dampened. And that really reflects the gene expression profile. So there's a disruption of the mitochondrial rhythms in myotube cultures from people with diabetes. So to try to get to some of the mechanism, what we did was we were looking for genes that are important for regulating mitochondria. And one of the genes that came out in an analysis of biopsies from healthy versus diabetic people is an alteration in genes that are associated with inner mitochondrial membranes, and one of those genes is OPA1. So we contacted Antonio Zorzano, and he happened to have a mouse model that lacked OPA1 knockout mice. And we studied muscle from the OPA1 knockout mouse, and we can see that many of the clock genes, their expression was depressed in OPA1 deficient muscle. And what Brendan could do is he could go into the muscle cell cultures and he could silence OPA1 using siRNA, and that also led to changes in clock gene expression throughout the day. And so what we can conclude is that the clock amplitudes, the amplitudes of these clock genes are depressed in myotube cultures from people with diabetes. We can show that there's alterations in the mitochondrial rhythms in myotube cultures from people with diabetes. We can show that, in experiments I haven't shown here, but you can read about here, the clock proteins, they bind to the inner mitochondrial membrane promoters of genes. And so there's a relationship between the clock and the transcriptional regulation of genes that control the mitochondrial rigor. And if we disrupt genes that are important for the inner mitochondrial membrane, we can disrupt the rhythms of these clock genes. So there seems to be a bidirectional feedback between the clock and the mitochondria, and this is something we see in people with diabetes. So where are we going? What are we working on? So what we're really interested now in working on the question of energetic stressors, diet or prolonged fasting, and we're wanting to ask whether or not energetic stressors, exercise, can influence the clock and control metabolism. And can we achieve this through energy sensors like AMPK, or nutrient sensors like mTOR, or oxygen sensors such as HIF1 alpha? And we have evidence that HIF1 alpha may be one of the transcription factors that contributes to the orchestration of these changes. And so the idea would be, if we silence these genes in cells, do we alter the clock? Does that somehow then feedback on metabolism, and can exercise restore this dysfunction? All right, so to summarize, what I've shown you is that exercise training can enhance insulin sensitivity, it can improve blood glucose control, even in people with type 2 diabetes, and there may be a better time of day to improve your glycemia if you have type 2 diabetes. So I've shown you also in animal models that the time of day can specify the rewiring of metabolic pathways and systemic energy homeostasis. And we can show this in mouse models. We have some evidence as well in humans. I've shown you that obesity, weight loss actually restores some of the changes in clock gene expression. I've shown you that free fatty acids and diabetes can influence skeletal muscle clock gene expression and clock output genes that are associated with metabolism and development. And then finally, I've been able to provide evidence where we can link the inner mitochondrial membrane rhythmicity to the circadian clock machinery. And we can show that this link is disrupted in muscle cells from people with diabetes, and that is associated with impairments in oxygen consumption rates. And so my last slide are the teams, and as you've mentioned in the introduction, I'm working between the Karolinska and the Center for Basic Metabolic Research, and I want to thank my team, because without them, I would certainly not have a whole lot to talk about. So thank you again for this invitation, and we have time for questions.
Thank you, Juleen, for this great presentation. So we have probably many questions. Let me start for one question. You basically said that in the afternoon the mitochondrial proteomes will change in response to exercise, and in the morning, if I recall, the glucose metabolism proteome will respond to exercise. My question is related to the fasting that we can have overnight. If the length of this fasting can influence the response of and the beneficial effect of exercise. Because there are a lot of literature saying that it's good to do a big fasting during the night, or sometimes it's good to have breakfast. So what is your opinion on that?
All right, so I clearly think we would need more data, particularly in people who are type 2 diabetic or people who have obesity. We would need more data. But I think there's evidence from healthy individuals that prolonged fasting can actually boost up the mitochondrial machinery. And so that might be an advice, that if you want to improve your endurance potential in a cell, at least, the fasting, the prolonged fasting, may have a beneficial effect to boost up the mitochondrial machinery.
So do we have questions in the room? Oh, because I have thousands of questions. So,
thank you.
Another one concerning the... okay. Okay.
Thank you for your presentation. On one slide, tell me if I'm wrong, but you show us that performance was greater in the active phase, but effects on health was different, was better on the other phase. I think it was the difference.
So I think you're referring to the rodent study, is
Yes.
that right? And in that study, there was always a greater effect of, well, the rodents are nocturnal. So basically when they're running during their active phase, when they're normally meant to be exercising, there is where we found the greater effects on metabolism and systemic homeostasis. When the rodents were exercised during the light cycle, when they're meant to be sleeping, we didn't get much of an effect on the metabolic outputs. And we had these rodents run on a treadmill. So Gad Asher has some data that there may be differences in the ability of the animal to perform work. And so it might be that they were having a difference in their work output. We could test that by looking at lactate. But at least in the mouse study, there was a clear separation of, Zeitgeber 3, which is the light cycle, the early rest phase, and Zeitgeber 15. I don't know if I answered your question, so.
Thank you, Professor Zierath, for this great talk. I have a question about the combination of exercise and intermittent fasting. Actually, diurnal and nocturnal intermittent fasting are proposed to mitigate metabolic disease, which is the most beneficial intervention, combining exercise at different time of day and intermittent fasting, diurnal or nocturnal intermittent fasting, when taking into account the circadian rhythm of medication. For example, for type 2 diabetes, the timing of the anti-diabetic medication.
Okay. So your question is related to chronomedicine and taking that into consideration along with chrononutrition and chronoexercise. And I would say, at least today, to go to a population of people who have diabetes or obesity, I wouldn't give that prescription yet, because what's probably the most important thing for all of those considerations, particularly with the medicine, is to make sure you do take it. Surprisingly, many people who are suffering with diabetes don't take their medicines. And so it's very important to take the medicine. And then I think we could get into some studies where we can assess whether it's better to take the medicine in the morning or the afternoon and how exercise affects that. And, the best thing would be to do these studies in humans and to do some clinical trials. That would be the best thing. But of course, one could do some studies in animal models as well. But I'd like to see more clinical trials where this is tightened out. Now, in the cancer field, they have come closer to helping us understand chronomedicine better. So some therapies are better if they're received in the afternoons versus mornings, for example.
And for the difference between diurnal and nocturnal fast, which you recommend, diurnal or nocturnal fasting for human?
I think your question is related to intermittent fasting. Is that right? I don't hear the question so well.
If we would propose an intermittent fasting program for diabetes people, which is the right timing of fasting? Is it diurnal fast?
So I don't know that there's enough data out there to give you one answer. So what I'm starting to understand more and more is the personalized approach to this as well. And so we can make one recommendation for the population, but it might be very different based on an individual's chronotype and habits. But there's some evidence that prolonged fasting can have an effect to move the cells to rely more on amino acid metabolism. And there's been some studies where prolonged fasting or intermittent fasting has no effect at all on metabolism. So I think we're still at early days, and we need very carefully controlled and large studies. And then the outcome should be defined. What is it that we're trying to improve? Is it glycemia, for example, or is it losing body weight, for example? So there's many things to think about still.
Thanks.
Actually, I can see you very well, so please manifest yourself.
Thank you, Professor, for this great presentation. So my question is about the study in which you try to see the optimal training time of day for training to reduce the peak of glucose. So when we see the slide, it was clear that during the first week, training during the afternoon is much more better. But during the second week, I saw that this difference was already reduced between the morning and the afternoon. So did you think that maybe we should test longer duration of training, like 4 weeks, 6 weeks? So maybe this difference will be blunted after 4 weeks of training or 6 weeks of training?
Right, so that's a really good observation. So what I showed you was the continuous blood glucose monitoring for week 1 and week 2 to the time of day of exercise training. And this was a pilot study. And frankly, I thought when we performed the study that we would get a blood glucose lowering effect of exercise irrespective of the time of day. So we were very surprised to see the hyperglycemia with the morning training. And if you notice, when we looked at the composite for that blood glucose monitoring, they had higher blood sugar before they even came into the lab, if you saw that. And I think there may be behavioral considerations. It might be there is a greater stress on the individual who's not used to exercising to have to get up early, to come to the gym, to participate in that training. And they become accustomed to that, so that at the second week, we didn't see such a profound effect on the glycemia. There was actually an improvement. So I would think that once an individual is routined and they're more familiarized with the training, that may go away. And so clearly we need to study that more. But what is interesting is this elevated glycemia in the morning, and Michael Kjær from Denmark in 1990 published a phenomenon very similar. And he speculated that that was a greater stress for the person with diabetes, and that over time, they become adapted to that.
Concerning your last part of your talk, I have a question on your myotubes in culture. So you show nicely that the mitochondria cycle in the normal myotubes, and that in diabetes it is disrupted, this cycling of respiration. Did you try to electrostimulate your cells to see if you can reset something in the diabetes cells?
Well, so what I can say is that all of those cells, the diabetic cells, you could call it, and the healthy cells, they were synchronized. So they've got that shock. And so there's probably a deficiency in some of the machinery that promotes that mitochondrial biogenesis. And so the experiment would be maybe to exercise those cells, a couple times a day in a dish with electrical stimulation, and see if you can overcome that. I showed you work from Nico that exercise did boost the mitochondrial biogenesis. And so I would think that regular exercise may help the person with diabetes overcome some of those mitochondrial defects that you reported.
Thank you. Another question?
Thank you very much. for your nice talk. I have a question about the rhythmicity of the oxygen consumption. So you said that it was kind of related to mRNA increase in mitochondrial protein, but you also involved OPA1. Do you think it's mostly, this rhythmicity is mostly induced by an increase in protein in mitochondrial biogenesis, or maybe more mitochondrial dynamic change?
I mean, I would only speculate at this point because we haven't studied that. So what we studied in those cells was mRNAs, and we haven't looked really at the protein abundance of those cells. So we would have to look at that more. So at least based on the data, it's gene expression, but it might be more mitochondrial dynamics as well.
Other question?
Well, I'll be around, and if people have other questions, they can come up and chat with me afterwards.
So thank you, Juleen, for this nice talk.